First of all, I would like to thank the organizer for the opportunity for me to stay here for this important meeting. Okay, let's go. I think that the famous sentence of the French philosopher Auguste Comte about the science and history of science help us to understand better the path and progress of the studies of the origin of life, beginning from the famous book of Alexander Oparin, and particularly mainly after the English edition of the starting from the first observation of Oparin, a lot of different hypotheses have been proposed to explain the origin of life on Earth and on the nature of the act in which life grows. But while most researchers agree on the fact that life appeared after the cooling of the Earth and its crust solidified, this thing is not a very habitat. Waiting for next Friday and the lecture of the short of this argument, I think that it is not a trivial argument, it's a fundamental aspect of the origin of life. In fact, also Charles Darwin, in the famous letter to his friend, the botanist and James Speaker, indicated that I would like to outline the sentence, but if I don't know what to believe, if today we do not share the same pessimism about pessimism of Charles Darwin about the possibility to explain the first stage of life, still the famous sentence of Erwin Schrödinger, What is Life? The famous book of the question raised from Erwin Schrödinger still remains unanswered for the difficulty we have in defining a precise boundary between non-living and living matter. One possibility to solve this problem is that to look at living organisms. All living organisms on Earth share the same program based on nucleic acids and protein. The genetic information recorded in DNA and RNA are necessary to produce, to obtain, to synthesize the protein. And the genetic material is handed down by a replication process from one generation to the next, so ensuring the perpetuation of the genetic information. This is the fundamental aspect of life. And even if other features are important for life, I think that the genetic information is absolutely essential. As pointed out in a paper of my friend Antonio Lascano on Oparin 2010, where Antonio outlined the importance and the present effect current biology indicate have not evolved in the absence, and the evolution is the fundamental aspect of life on this earth, on this planet. So, if we look at the molecular evolution of this planet, we can say that in the era, ancient era, the appearance of a nucleic acid polymer able to evolve from the Darwinian point of view, in my opinion, but not only in my opinion, marks the beginning of life. genetic polymer could originate. The formation of a genetic molecule, whatever it was, would require specific conditions, including the synthesis and the combination of precursors, the polymerization, the joining of these simple molecules to obtain larger molecules, their protection from degradation. This is, in my opinion, important aspect, because the situation at the beginning of life is not so easy for life, because we have a shell of ozone and effects. This protection is important for ensuring to genetic polymer the possibility for to express their biological potentiality. On the basis of the knowledge of the 30s, last centuries, the ideas of Oparin was based on specific intuition about the composition of the atmosphere, the so-called the primitive or reducing atmosphere, was composed mainly of anoxys gases and anoxys gases and water vapor. The genesis, John Haldane, a few years later, suggested that the simple molecules present in the primordial atmosphere under the effect of specific energy, sparks and other things, could react and to form simple molecules that then floated in the primordial oceans, the primordial soup, the so-called primordial soup. But experiment, famous, the pivotal experiment of Stanley that demonstrated the validity of this hypothesis. However, as biological polymers are formed by the rotation in general, it is difficult to conceive that these complex molecules can be originated in primordial ocean, that is in the presence a high concentration of water, because in this concentration we have hydrolysis and not polymerization. This is particularly evident, as Professor Spearing and other people this morning indicated. It is particularly evident in the case of RNA work. The RNA molecules, which is supposed to be the first genetic material appeared on Earth, waiting on my colleagues, Marie-Christine, in the next presentation. We can say that the presence of the oxygen group renders the molecule particularly unstable in water solution. It was John Barnard that first indicated, suggested the clay minerals, or minerals in general, a fundamental role of clay minerals in promoting the chemical evolution that then the origin of life. In fact, in a famous book in 1951, 2 years before the results, the science article of Miller indicated that the clay omicron were necessary to concentrate the organic present in the primordial ocean, to protect the molecules from destruction by harsh radiation and to catalyze the polymerization of these molecules. Clay minerals were present on the surface of Earth. Clay minerals originated in the weathering, volcanic glass and rocks. And now, at present, results that from astrobiology field indicated this fact. The Mars investigation, in fact, indicated the occurrence on the surface of a red planet of a clay mineral with an age of about 3.5, 3.6 billion years ago. By and large, clay minerals would have formed on our planet. This is a picture taken by NASA. The green color indicates the presence of clay minerals on the surface of Mars. This is now demonstrated. The role of minerals, mainly clay minerals, in the synthesis of components of nucleic acid are demonstrated in numerous studies, particularly in the production of nucleic acid bases by the group of Saladino and Ernesto Di Mauro. And also, I remember a good paper on the importance of the presence of minerals for stabilization of ribose, the sugar present in our RNA. This is now confirmed by other studies of mass composition. With regard to the polymerization, studies carried out by my friend Jim Farris at the Rested Polytechnic Institute and his group indicated that these the famous results on the end of the 90s on the polymerization on clay minerals surface up to the polymerization oligonucleotide up to the length of a short ribosome. Studies done by the group of Matthew and Lute Schutt by using classical molecular dynamics methods indicated that indicators seem to suggest that the organization of nucleotide take part in the interlayer between the different layers of the clay. It is just I know. So, as Judith can be explained, this fact that it is not. But at least we have these results. With regard to the protection of nucleic acid, I would like to describe some words because these results I obtained many years ago, before at the New York University, and then in my group in Florence. Results on bacterial genetics, molecular bacterial genetics, indicated that the DNA molecules absorbed on clay minerals are protected to some extent against the action of different degrading agents, but more important, still maintain their biological activity, as demonstrated by the ability of clay DNA complexes to transform competent bacterial cells, and also the DNA that it's also able to be recognized by different enzymes. For instance, we found that DNA absorbed of clay can be replicated and amplified by polymerization reaction. These very old results pressed us to study in detail the molecular characteristics of a clay DNA complex. We analyzed this complex by different methods. For instance, you can see here the picture of a transmission electron microscopy. You can see different fibers of DNA that are connected with the some point of the edges of clay particles. And it seems that this is a plasmid DNA, it seems that the DNA can lie directly on the planar surface of the clay minerals. To understand better the process of absorption, we studied also, we started to analyze the complex by X-ray diffraction analysis, and the results we obtained indicate that we have no, intercalation. That is, the molecules are absorbed on the surface of a clay. Not only we investigated the molecular characteristic of a clay DNA complex by Fourier transform infrared analysis, and we observed that when DNA is absorbed on clay, it changes its characteristics, molecular conformation. We found that when the DNA was absorbed on clay surface, it passed from the B form to A form. The A form is the resistant form of DNA. The form of DNA we found in the mineral in the outside cell. During the process of absorption, cations, positive ions, played a fundamental role because of the cations, monovalent and divalent cations, act as the bridge between different negative charges present on the backbone of the molecules, and the negative charge of the surface of the clay. But more important, we found that when the DNA is absorbed on clay, it's much resistant, is protected, is much resistant to the effect of ultraviolet and X-ray radiation. In fact, we found that the percent of dimers of thymine in thymine dimers is reduced when the DNA is absorbed locally. This observation are recently confirmed by experiment carried out on board the International Space Station during the last flight with the use of a shuttle. We sent DNA absorbed on clay, and the DNA was exposed to the cosmic ray. After the return, we analyzed the DNA and we found the fact that the only DNA absorbed on clay maintains its integrity. We extended also our investigation on RNA complexes. Different RNA molecules were absorbed of two minerals, clay minerals, montmorillonite and carbonate, like DNA complex, and the complexes were analyzed for different aspects. For instance, we found that single-strand polyomnionic RNA, in this case poly-A, are linked specifically with the corresponding poly-U chain. But the RNA absorbed on clay still maintain the ability to be recognized by PCR technique. In this case, the reverse transcription PCR technique. But also, we extend our investigation to the studies if the complex still perform an enzymatic reaction. For this studies, we utilize two different ribozymes, hammer-head ribozyme and hairpin ribozyme. Specifically, we utilize the hammer-head ribozyme present in Adultatus ambloch viroid from the group of the Flores in Valencia, and the other is the adenine detected. Hairpin is a gift from my friend Marie-Christine Morel, We studied the interaction of ribosome with the clay particles. In particular, we studied the ability to perform a self-cleavage reaction and to resist to the action of various degrading agents, and demonstrated by these results that we published some years ago on the resistance to UV irradiation. So, on the basis of these results, we can suppose that the clay-rich sediments would be a good habitat for the origin of an RNA clay world. In this situation, when molecules dispose of rocks in contact or in the presence of phosphate outside, and also the solution contains small molecules, we can have the polymerization with the formation of the molecules, not only the formation of the molecules, but also its life in this condition, under also difficult conditions presented by, for instance, radiation. In this case, the molecules inside can evolve, can interact, triggering the molecular evolution that led from the first Klein cell of Schost group to the present living cell. In my opinion, these results are not sufficient, but are important to at least to solve the famous myth of Prometheus on the origin of man from mud. Before concluding my presentation, I would like to remember the people who have passed studies of different aspects: Professor Ferris, Riccardo Flores, Marie-Christine Morel, and Professor Dante Stosky at the New York University. They began the studies of the EVA before finish. I had to thank one person, a special person present here in this room. I think to interpret also you think about to thank my friend Misha for the work that does to stay here in Moscow. Thank you for your attention.